This is Holy Cross Component 1 mock, and it's mock number 3. Question 2. And here we've got a growth curve of a colony of bacteria. And it's our standard looking growth curve. And initially we've got it from the serial dilution, and they just want to know how you carry out a serial dilution. You've done this in the lab, pretty straightforward procedure, and it's just doing the same dilution each time, and that's what the serial mean. Yeah, a serial killer would do the same thing each time. So we take a known volume of culture, and what you've probably done is 1 cm cubed, and dilute with sterile culture medium, e.g. 9 cm cubed. I would definitely say that rather than water, and certainly even if I was saying water, I'd say sterile water, because you always want to avoid contamination. And then take the same volume again, and repeat that in the next sample. And you remember this, so you've got tubes. The tubes have got 9 of sterile culture medium. Here's our original culture, and so we're taking 1 out of there, mix, then 1, mix 1, mix 1, and so on. So repeat again and again to give a series of higher dilutions. Why is this necessary? Well, if we took our original sample out of our original culture, made a petri dish and grew it, well, it wouldn't give those nice colonies that you would count like that. It would just look a mass of colonies that you can't count. So the number of cells in the original culture is too great to count. And here's a little bit of maths. Concentration of bacteria in a sample after 4 tenfold dilutions was 85 per mil, or 85 per cm cubed. Calculate the total number of bacteria in the original culture if its volume was 20 mil. Okay. And so what they are saying is that we've done exactly like we have in the diagram here. We had 20 mil of our original culture, and we've done this dilution 4 times. And so what we've got is our number 85. We've got to times it up by how many times we diluted it. So 4 tenfold dilutions. So it wasn't 10 times, it wasn't 100 times, it wasn't 1,000 times, it was 10,000 times. So there's our No, original, 10, 100, 1,000, 10,000. And then that would give us the kind of concentration, but it says how much have we got in this entire 20 mils? So we'd have to times that by 20 again, because we've only counted it in 1, and we need to times that up for 20. And so when we do that on our trusty calculator, 85 times by 10,000 times by 20 gives us this number here, which is 17, and then it's got 1, 2, 3, 4, 5, 6 zeros. What units would we have? Total number, it's just total, so the units is just bacteria. You might have represented that in standard form, 1.7 times 10 to the 1, 2, 3, 4, 5, 6, 7. Should have turned my phone off. What is the advantage of viable counts over total counts? Pretty straightforward if you're doing a revision. The total will give you all of the cells, living and dead. The viable will only, because it's based on growth, the only ones that will grow are the living ones. So viable counts don't include dead bacteria. Or you might have said it only grows the kind of living bacteria. On the next page, name the 4 phases. That's okay. We have got on here, we got lag, exponential, or sometimes called the log. That's the stationary phase, and then a decline or death phase. So we just need to write those out. Got, it just says lag, it just says name. So lag, we've got exponential, stationary, and death. So it's just naming them. Explain the shape of the graph. So hopefully you've, in your notes, you've just got the name of them, and then you've got a sentence of explanation, and you can just trot that out here. In the lag, we've only got a few cells present, and so even if they were doubling, you wouldn't notice it very much. Or they might be acclimatizing, not reproducing very much. It might be, switching genes to make use of a new medium, that sort of thing. So few individuals present, or we could say, very low reproduction. Then when we're on to phase B, we can say, well, we've got a maximum rate of reproduction. We're not just going to say exponential again, because we've said that in the name. Birth rate's higher than death rate. We've got lots of resources, nutrients, no limiting factors, that sort of thing. So if I put down no limiting factors, e.g. nutrients, and maximum rate of reproduction. Be careful saying birth rate and so on, because these bacteria aren't born. They reproduce by binary fission. Other things you might have said there, you might have said, they double in a certain amount of time to these bacteria. And in C, well, that's our stationary phase. We can say the production of new cells equals the death of old cells. We've reached our carrying capacity. Carrying capacity is reached, and our resources are now limiting. So it might be nutrients or oxygen are limiting now. Our rate of production of cells is equal to our rate of death of cells. And then in the death stage, we've got depletion of nutrients or resources. So depletion of resources. We can say that the death rate is much higher than the production of new cells. And we could say toxins accumulating. That sort of answer. So we can say death rate much higher than reproduction. And then a little strange end to this question: if a continuous culture, one where you'd instead of having it just in a flask like that, you have it in a kind of big fermenter where they're constantly putting medium in and constantly taking the bacteria out, and so it's just goes on forever and ever rather than finishing. Which of the phases would be best source of this sample? Well, even if you didn't know what a continuous culture was, where would be the best thing to get cells from? Well, it'd be this exponential phase where the cells are happiest. So the phases, let's go for exponential, or if you got that wrong, you might have said region B on the graph. Give a reason. It's got the most rapidly reproducing bacteria that are now acclimatized to the medium, less likely to have dead cells there, those sorts of things. So rapidly reproducing viable bacteria. So 15 marks, but quite a lot of it was stuff that you just had to remember from your notes.